Hi, thank you for the introduction. I'm very pleased to be here. I'm a native Romanian, left quite a while ago, but I'm really happy to be back in Romania and give a talk here. Thanks for the organizing committee for making this possible. So I'm currently based at the University of Vienna, and one of our research interests are adaptive radiations. These are, represented by clades, which are highly speciose, and in these clades, lineages normally find and occupy different, and so very different niches, ecological niches, and they adapt differentially to these niches. You can see an example here so I will talk today about the adaptive radiation of persimmon trees in New Caledonia. Most known examples of adaptive radiations come from animals, actually, such as cichlids in East Lakes in Africa, or that Darwin finches, right? These are classical examples, and the classical theory suggests that such adaptive radiations actually are made possible by ecological opportunity. So this is seen as the main driver of such adaptive radiations, ecological opportunity. Normally after long-distance dispersal events, right, a wide range of available niches, unoccupied niches, become occupied by these lineages. Anyway, recent studies on such animal systems normally, because they were mostly studied up to date, show that the story is not so simple. So we cannot invoke just ecological opportunity to explain these adaptive radiations, and other processes such as hybridization or demographic events, or even genome evolution may also play a part in these processes. And so we are trying to ask these questions if, altogether genomic potential driven by, events such hybridization or genome evolution need to be invoked when we are trying to understand how adaptive radiation proceeds, even in plant systems. And we'll talk today about persimmon trees on New Caledonia. First, a few words about New Caledonia. This is an archipelago 1,600 kilometers east of Australian coast in the Pacific Ocean. New Caledonia became, split off Australian continental plate a long time ago, 80 million years ago or so. And then it became submerged underwater, and it was covered by oceanic sediments, which are heavy metal rich. So after its re-emergence, roughly 30 million years ago, these oceanic sediments gave rise to ultramafic and serpentine soils, which are very difficult for plants to adapt to because they have a high heavy metal content, but also a skewed nutrient content, and also they normally have low retention of water. In addition, after this re-emergence in the last 30 million years, there was a lot of erosion. So New Caledonia at present, it's a highly heterogeneous environment with very patchy soils, so very close by very different soils. And in addition, it has a mountain range in the middle of the main island, Grand Terre. And you can see it has very sharp slopes normally. And rain and water, wind and rain come from the eastern side, so there is a wet side on the northeastern side, and the dry side on the southwestern side. And all together with these climatic differences and the differences in soil, New Caledonia became a biodiversity hotspot, and it's currently cited as the area with the highest level of endemism in the world. Here are some examples of the habitats. You can see in the upper row, for example, humid forests on ultramafic substrate, dry maquis on ultramafic substrates, littoral forests, or here dry forests on sedimentary soils. And some of this vegetation is highly threatened by mining and introduction of mammals, of course, deers and rabbits. And some of them are highly threatened. For example, dry forests. It's estimated that just 1% of the initial area occupied by dry forests remain on the island at present. So there is a high need for conservation and actions on the archipelago. A few words also on persimmons. So Diospyros is a large genus. It includes perhaps more known members such as edible kaki or persimmons, which you can find in supermarkets, and also ebony timber. But the radiating clade on New Caledonia, it includes more than 30 species, and this originated after a single long-distance dispersal event. It has diverse morphology. You can see the variation in habitat. In habitus, Diospyros sharieri forms large trees, whereas new species in Ile-de-Pin are just shrubs of 1 meter high, and they are adapted to this very dry desert, almost like ultramafic niches. So diverse morphology, also leaf shape or size. You can see here the two perhaps extremes, and also very diverse morphology of a persistent calyx on the fruits, which is actually one of the main characters to recognize the species. The trees are dioecious, and most of them have a specific ecological preference, and they occupy actually all habitats on New Caledonia. So you find them really everywhere apart from mangroves, so this is the only habitat where they don't occur. A few years back, we had a data set of roughly 80 accessions, and we used rat sequencing to try to solve the phylogeny of the group. So this is just the radiating clade on New Caledonia. First, you notice that all these speciation events lead to different ecological preferences. So sister species are normally occupying different niches. In the colored column, you can see the different substrate preference, and in the gray column, you can see the preference for climatic conditions, dry versus humid. You can see that mostly the climatic conditions separate the nodes which are deep inside. So the first nodes, for example, Vailardi is growing in dry climates, whereas the next clade is growing in humid, whereas the latest speciation events are normally leading to different substrate types. We estimated also that the radiation is roughly 1 million generations old. The generation time is roughly 9 years. But actually most speciation events are in this group 2. So roughly less than 500,000 generations. So this is a young radiation. It's quite speciose, and it's also atypical because normally radiations you expect an explosive speciation rate at the beginning, and then this speciation rate should go down, but we see the opposite. So we think that it's still undergoing speciation at the moment, and actually we see with demographic inference, which results I will not show today, that they still go through population expansion, or at least the signals are that it's going through population expansion. I said that we gathered this data set to look at the phylogenetic relationships, and some of the relationships are not well resolved. So now we went back to New Caledonia last November, and we started to collect a lot of data, ecological data, soil chemistry, leaf chemistry, metabolomics, and so on and so forth. Less than 1 year ago. So I'll go slow. I will not present you everything because most of these results are not available yet. We have more than 350 accessions with whole genome sequencing. You can see some of the results here. We are looking now at the basic information, for example, cryptic speciation, with such examples as you see here. Perplexa, for example, has a clade here and one here, and so on and so forth. We are also using this data to run demographic inference and also to look at introgression. And I wanted to look in particular today more in the introgression. So we run these statistics and we observe a low but pervasive hybridization actually among almost all species pairs in the radiation. You see this heat map shows you the results of this statistic. If it goes to dark red, it's more stronger evidence for hybridization, whereas if it goes to stronger colors, the significance it's higher. And you see that, yeah, so there are some areas where there probably it's either a more recent event or the evidence is stronger, but actually most of these cells are statistically significant. The same we get when we do other methods like tree mix. For example, you see a lot of connections which are needed on top of the maximum likelihood tree to, account for the data. And also with methods like HIDI, which is another statistic related method, in which we looked in particular for species pairs which share the same habitat, but or similar habitats, but they are not so closely related. So there is a lot of potential here to look more into the local genomic landscape of introgression and see which genes may be affected and flow between the species. So we think that, yeah, hybridization is definitely, because of its prominence, right? We think that hybridization is definitely one of the factors contributing to this radiation. In addition, I did not mention this up to now. So on New Caledonia, there are 4 lineages of Diospirus actually. And just this one, which I talked about, it's undergoing adaptive radiation. You can see it here in this green clade. So this is the sketch of the phylogeny of the genus. And the other colored clades, Samoensis, fasciculosa, and here, are the other 3 lineages which arrived on New Caledonia after long-distance dispersal events. And the interesting question is, what is different between the radiating clade and the other clades, and how can we, explain this radiation? One of the main differences is actually genome size. So the radiating clade has almost twice the genome size as the non-radiated lineages, but this is at the homoploid level, so these are not polyploids, and they are linked to TE dynamics, which increase genome size in the radiating clade. We have these are early results based on genome skimming, in which we observed already a high variation in transposable element content in the radiating clade. Now with the new data, we look again at these kind of aspects where we observe, for example, you see here a heat map of TE content as counts per million of short reads mapped to a consensus sequence of transposable elements. And in rows, you have transposable elements families or consensuses. And in columns, you have representatives of the radiating clade here. And yeah, just very shortly, there is differences in enrichment of one or the other classes of transposable elements. In particular, the LTR transposable elements in the gypsy family seem to be overrepresented in this clade, which are is highly variable and highly present in this radiating clade. The same we get when we look at reference genomes. We have draft reference genomes of several species, and we are looking, for example, here comparing one species of the outgroup. This is Sandwichensis, a Hawaiian endemic, and one species from the radiating clade here in green. And you can see that the branches of the different copies of this LTR copy, so family 2, for example, are, so the branches here are very short, showing that there is a recent expansion of these transposable elements in the radiating clade. I'll skip this. So the summary, we have 3 ideas here for something. So the radiated Diospirus definitely experienced highly reticulated evolution with both ancient and more recent gene flow visible, but we have to look into the local genomic landscape of introgression to see if we can spot an involvement of hybridization in phenotypic trait shifts. Then T dynamics are associated with this adaptive radiation and not in the other lineages. And we still need to answer if this is stimulated, so this T dynamics is stimulated by hybridization and if there are links to adaptation. But definitely, ecological opportunity alone is not sufficient to drive adaptive radiation. Also in this plant case, because there are 3 other lineages which did not undergo radiation with the same opportunities. So some acknowledgements, and thank you very much for your attention. Happy to take questions.
Thank you very much. That was a really interesting talk and some beautiful photos. Has anyone got questions? Yeah. Bertold?
Thank you. Thank you, Ovidiu. Congratulations, very interesting work. Thank you also for mentioning Mayra Brisenu. She's now in my group for a limited time. The question I had in connection to one of the previous talks, if you know the timing so well of the radiation, do you think you could estimate, or maybe you've already done, estimate the mutation rate for these glades that would enable to see the differences in genomes that you now see, and would that how would that compare to plant mutation rates that other people have estimated?
Thank you for the question. I'm not sure. Yeah, we could try, definitely. This would be a very interesting idea. Actually, we have a proposal actually submitted right now for this. I'm not sure. We'll see what the funding agency will say. But however, I definitely would like to estimate the time of these TE expansions. Because there are several hypotheses. One would be that they are linked to the bottleneck, the initial bottleneck event, where selection was not so efficient. And another one would be that this is continuously happening across the radiation. And I think I'm very interested in that question right now. But yeah, we have a plan for mutation rates as well. Thank you.
Hi, Ovidiu. Thank you so much. I was wondering if, regarding this expansion in TE families, if you also observed some interesting insertions in gene coding regions, maybe with some connection to, ecological factors.
So I don't know yet. I mean, this is the hope. Our draft genomes are not yet very, very good to look into. I mean, yeah, we started to look into annotations and so on, but this is a non-model and very horrible chemistry. So, but we are getting there. So, yeah, maybe give me 6 more months or so.
Okay, I think we've maybe got time for one more question. Is there anybody online who is wanting to ask anything?
I had a one here.
Oh, Tanya.
So, kind of similar question as the previous question, but what's like about the connection between the TEs and the radiation? So, is there some mechanism how TEs could like promote development, creating reproductive isolation, or just make speciation happen more easily instead of just facilitating adaptation?
Yeah, thank you. So there is a lot of literature already on this. Slowly getting to switch from these detrimental effects of TEs to the positive effects on speciation or so. TEs could be involved in triggering other structural variants through illegitimate recombination, for example. They offer the sites for that. And then such translocations on inversions are known to be linked to speciation. And also they can exonize, or they can silence genes, they can become regulators of genes. They can have a lot of effects at the phenotype level. Many of them detrimental, there is no doubt, but obviously, or we think that there should be also some positive effects.
Okay, thank you very much again. So now we're going to have... Oh, sorry.